Hello everyone. Welcome to Inspire, a short lecture series in science. In short videos, we discuss many scientific facets. Each video addresses one question. Today, we are going to learn about cells, the building blocks of life.
When and where were the cells first discovered? How did they get their name?
In circa 1650s, a scientist named Robert Hooke observed a peel of cork through a lens. He noticed a regular arrangement of some structures, much like stacked boxes, and it reminded him of rooms in a monastery, and he called them cells, and the name stuck.
So, you said these cells were the building blocks of life. How do they build up living things?
Well, groups of similar cells make up what is called a tissue. Animals have four major types of tissues: connective, epithelial, nervous, and muscle. Groups of tissues make up organs that perform a specific physiological function. Examples are heart, lungs, or the brain. These in turn make up organ systems such as the vascular system or the digestive system. These systems finally make up an organism. The cells are the actors behind all biological activity, and that is what we will see in detail today.
So, before we go into the details of a cell, I have some very basic questions. One of them is, how many different types of cells are there?
Well, there are innumerable types of cells, but to begin with, we focus on two major groups, that is the prokaryotic and the eukaryotic cells. These cell types represent two fundamentally different branches of life. The word karyote is derived from the Greek word karyon, which means kernel. In this case, it represents the nucleus. Prokaryotes are those that lack a nucleus, and those other subcellular membrane-bound organelles. Eukaryotes have a true nucleus and a host of other membrane-bound organelles. In eukaryotes, the genetic material is present within a membrane-bound organelle called the nucleus. However, in prokaryotes, the genetic material is generally concentrated in a particular region of the cell and it is not surrounded by a nuclear membrane. This is called a nucleoid.
So, there is another basic question from my end. How big do these cells get?
Well, bacteria are typically smaller than eukaryotes, understandably, and they are about 1 to 10 micrometers in length. Eukaryotic cells could be anywhere between 10 to 100 micrometers in size. One of the largest single eukaryotic cells is the frog xenopus egg, which is visible even to the naked eye and could be up to 1.1 millimeters in diameter.
So, this is a pretty large cell. That's very surprising. I'm guessing the size of the cells will also depend according to the type of the cells.
Of course, the sizes are as diverse as their shapes. We should first know about the different types of cells themselves. In a typical human body, we have various different types of cells, such as the red blood cell, as you can see, the disc-shaped cells. Then you have the immunological cells, such as the polymorphonuclear cells or the white blood cells. Then you have the neurons, which have long axons, which could be meters in length. Then you have the muscle cells, such as the smooth muscle or the cardiac muscle cells, and the epithelial cells, and so on.
This is very interesting. I see a wide variety of shapes. I see the disc-like shape of a red blood cell. I see this amoeboid shape of a white blood cell, the box-like structure of the epithelium, and the really indescribable shape of a neuron. How do they tend to keep these shapes? How do they maintain these shapes?
Well, that's a great question. They have an interconnected network of structural proteins called the cytoskeleton. The cytoskeletons are made up of three types of protein filaments: the microtubules or the tubulin, the microfilaments or the actin, and other intermediary filaments. Tubulins and actin are generally involved in support and transport. Intermediate filaments like laminins give shape to the nuclear membrane, for example, and then you must have heard about keratins that form the hair and nail. Eukaryotic cells can be of different shapes and sizes and even have different functions. However, they all have some common properties, which is an outer cell membrane that encloses the cytoplasm, the nucleus, and the other organelles. Plant cells, however, have a unique cell wall and chloroplast, as you can see in the picture.
So, can you now tell a little bit more about these components? That seems a very bewildering array of components within a cell, so it will be nice to know what each of them does.
Well, as I already mentioned earlier, the cell membrane or the plasma membrane, which encloses the cytoplasm, is a selectively permeable lipid bilayer that is interspersed with numerous surface proteins, transmembrane proteins, and transporters that form channels and allows the transport of substances across it. The structure of the plasma membrane is generally described as a fluid mosaic model that contains phospholipids, carbohydrates, cholesterol, and various proteins, which gives it a fluid-like characteristic. The plasma membrane or the cell membrane encloses the cytoplasm. The cytoplasm is sort of like a thick soup in which all organelles float around.
Is it a good comparison to say the cytoplasm is like a ocean or a sea and the organelles float on it like, say, much like ships and boats?
Yes, that is a good analogy. However, a better one is a pot of thick and crowded soup. Contrary to many depictions in pictures, the cytoplasm is actually a crowded place. Innumerable organelles, proteins, and other macromolecules are either suspended or dissolved in it, making it an ideal medium for biochemical reactions.
So you also mentioned, so we just covered the cytoplasm and the membrane covering the cytoplasm, but you also mentioned a lot of other organelles, right?
Well, we have already mentioned the nucleus. It is bound by the nuclear membrane in eukaryotic cells. The nucleus contains the genetic material or the DNA of the cell, which is present in the form of chromatin. The nucleus is the part that is generally visible on selective staining. During isolation in the lab, the DNA separates out of the solution like threads. The nucleolus is a region that is involved in the assembly of ribosomes, about which we will learn next. It is also involved in cell division. During cell division, the DNA is compactly packed into rod-like structures called chromosomes. The name comes from the way these structures absorb dyes and form a colored body. The DNA contains all the information needed to make proteins that are essential for the construction and functioning of a cell. When a cell divides, the genetic material is duplicated and each daughter cell or progeny receives a copy. Next comes the ribosomes, as I mentioned earlier. These are an important part of all living cells. Ribosomes are some of the oldest components known in living systems and are involved in protein synthesis. They are present in all living organisms, including archaea, bacteria, fungi, plants, and animals. The ribosome is a large nucleoprotein complex that has the ribosomal RNA or the rRNA. This complex reads the genetic information sent out from the nucleus in the form of messenger RNA or the mRNA. This information is arranged in a three-lettered code called the genetic code. Each codon corresponds to one amino acid, which is carried by the amino acid transfer RNA or the tRNA. The ribosome catalyzes the synthesis of a protein chain using the information in the mRNA and the amino acid in the tRNA. Without ribosomes, protein synthesis will not occur in biological systems.
So, do the ribosomes actually float around in the cytoplasm?
Well, in prokaryotic cells, they do. However, in eukaryotic cells, they are mostly associated with an organelle called the endoplasmic reticulum. These are membrane-bound network of tube-like structures that are often attached to the nuclear membrane. The endoplasmic reticulum with ribosomes is called the rough endoplasmic reticulum. They are the major centers of protein synthesis in the cell. The smooth endoplasmic reticulum, however, is mostly involved in lipid synthesis.
Lipids like cholesterol?
Yes, and a lot more. I earlier mentioned that the cell membrane is a lipid bilayer. Similarly, there are many other forms of fats and lipids that ensure our body works well. These are synthesized in the smooth endoplasmic reticulum.
So, what about the other sac-like structure that we actually see here, this thing.
Well, that is the Golgi complex or the Golgi bodies. This is where proteins undergo processing.
So, okay, let me get this. So, proteins undergo first, there is something called synthesis that is done by ribosomes,
Yes.
and it involves the nucleus sending out some information and then the ribosomes translating that information into proteins. But then there is something called modification or processing. What does it mean? What happens there?
Well, many proteins that are produced as such are not active. In order to make them active, either sugar molecules have to be added to them chemically or proteins have to be cleaved to make them active. This is called post-translational modification of proteins. Much of this happens in the endoplasmic reticulum and continues in the Golgi complex. Once the proteins are suitably modified, they are packaged into vesicles and shipped off.
Shipped off to where?
Well, either they can be shipped off to different parts of the cell or stored for later use or often secreted out of the cells.
So this processing of proteins, is it quite common in eukaryotes?
Yes, this is a very common process and many proteins undergo post-translation modifications. A well-known example is antibodies. After appropriate post-translation modifications, antibodies are secreted out. Another family of high molecular weight, heavily glycosylated proteins are the mucins, which is present in the mucus. Mucins make the mucosal fluid viscous and helps to lubricate the cell surfaces and are often involved in a range of immune responses. Even hormones such as insulin must undergo post-translation modification before they are activated.
Okay, so we have protein synthesis in the ribosome, we have their modification in the Golgi complex. What happens next?
Well, they can be either stored in membrane-bound sacs called the vesicles until they are used, or there are also storage organelles for other essential molecules known as vacuoles. Vacuoles are rare and generally smaller in animal cells, while plant cells have more prominent vacuole, which could be up to 50 to 90% of the cell. Plant vacuoles contains cell sap, amino acids, proteins, starch, etc. So all of the stuff that makes the cell eventually get worn out, and all the waste that is generated in a cell such as foreign materials, bacteria get digested or destroyed in an organelle called the lysosome. The lysosome is another vesicle which is involved in the degradation of proteins. They are sort of the waste disposal units of a cell.
So the name lysosome, I guess, comes from the root for breaking down lysis.
Yes, exactly. Lysis means to break down, and the organelle contains many enzymes such as lysozymes or proteases that break down the debris.
So what else do we have on the list?
I think we are left with two very important ones, saving the best for the last. The mitochondria, which is the powerhouse of the cell, and the plastids that are unique to plant cells. Mitochondria are perhaps the best known of the organelles, and it generates energy for the cell in the form of adenosine triphosphate or ATP. ATP is the energy currency for the cell. Mitochondria uses a process called oxidative phosphorylation to produce ATP. And of course, you must all have heard about the chloroplast, the green pigmented plastids of plant cells, which is the center of photosynthesis. There are also other less common pigmented plastids and colorless plastids called leucoplasts. It is not a stretch to say that without mitochondria and chloroplasts, advanced life on Earth wouldn't be possible. By the way, did you know that both these organelles have their own genetic material and protein synthesis capacities?
That's a very interesting factoid. I didn't know that. So let me just summarize what we learned today. We learned the basic types of cells, prokaryotes and eukaryotes. We learned that eukaryotes have membrane-bound organelles, subcellular organelles. We learned that ribosomes are some of the oldest and most universally found cellular components. We learned about the function of ribosomes in protein synthesis. We learned about endoplasmic reticulum, protein processing and modification in the Golgi complex. We learned about mitochondria and plastids and their role in sustaining life. So, with all of this knowledge, I fail to understand one thing. We know so much about cells, but where do we use them? For example, how do we grow cells in a lab? What do we use them for in an industry? And do they find any use in the clinics?
Yes, thank you for summarizing that. And scientists can grow cells in the laboratory in glass plates, petri dishes, or flasks in media that contains essential nutrients that mimic the natural environment of the cell. They study the various biological processes and phenomena. They use cells and tissues to see the effect of new drugs in the lab before developing them as treatments for patients. In the industry, cells have a wide range of applications, such as producing clinically important proteins, drugs, antibodies, and even viruses or antigens to make vaccines. Both prokaryotic and eukaryotic cells are useful in the production of industrially important enzymes. Many industries, including food, biopharmaceuticals, cosmetics, FMCG, textiles, tanneries, and even bioremediation processes, are heavily dependent on these enzymes. In clinics, we use cells to address emerging areas of stem cell therapy, in vitro organ development, and even in treating diseases such as cancer.
That was very informative, and I thoroughly enjoyed the discussion of cells, their role, and their application. I hope our audience learned a few things too.
I hope we were able to inspire you to learn more about cells. Do let us know if you have any questions or comments. Thanks and have a good day.
Have a great day, folks. See you later.